Hello! In this video, I'd like to talk about the important processes of meiosis and fertilization. Meiosis creates a huge amount of genetic diversity in sexually reproducing organisms, and this diversity is an evolutionary advantage for reasons that you've learned about in class. In this video, I'd like to focus on the movement of genes and chromosomes in these processes. So let's start and look at meiosis in an individual that has this genotype. Big A, little a, big B, little b. And let's just say, for this video's sake, that this is your dad's genotype. And we are going to, through this video, make the sperm that fertilizes the egg that led to you. So let's start in your dad with an unreplicated cell. And in this cell, the A and the B chromosomes, or the A and the B genes, are on different chromosomes. And your dad had to inherit these chromosomes from his parents. So let's say that your dad inherited the big A from his mother and the big B from his mother, and inherited little a from his dad and little b. So these are your dad's chromosomes that contain these genes in G1 phase of the cell cycle. Notice that his parents' DNA molecules are separate, and they are separate in every cell of his body. Now what happens before meiosis begins is that these chromosomes replicate. And as you saw in the previous video, semi-conservative replication occurs. Each DNA strand is used as a template to make new. So what you end up with is two identical sister chromatids held together at the centromere. So all I'm doing here is copying each of these. This DNA molecule has been replicated and is now two identical DNA molecules held together at the centromere. And the relationship between these two DNA molecules, they're called sister chromatids. So this is a replicated cell. Now before we take this cell through meiosis, let's look at how many DNA molecules and chromosomes are in each of these cells. So each line is a DNA molecule. So here we've got 4 total DNA molecules. And 4 unreplicated chromosomes. As you learned from the previous video, in this cell we still have 4 chromosomes. These are called replicated chromosomes. So we've got 4 replicated chromosomes and a total of 8 DNA molecules. Now this cell is ready to undergo meiosis. So the first thing that happens is that homologous chromosomes are going to come together. The cell recognizes that there are similar sequences between homologous chromosomes and brings them together. So I'm going to go ahead and draw that here. Is the beginning of meiosis I. So I'm going to bring chromosome with the big A together with the homolog with little a's, and the chromosome with the big B together with the chromosome little b. Now this is happening only in cells that are going to become sperm cells. So what we see here, the homologs are coming together, and what happens is that crossing over can occur. And crossing over specifically occurs between non-sister chromatids. And where and how many crossovers occur varies from meiosis to meiosis, and this can generate a lot of diversity. So what happens here is that a maternal chromosome, a chromosome inherited from your dad's mother, actually becomes part of the same DNA molecule as the as the homolog that was inherited from your dad's dad. So we can imagine that this is happening multiple times along the chromosome and differs from meiosis to meiosis, so you can create a lot of diversity. What you end up doing is shuffling the alleles along a single DNA molecule, so now that on one DNA molecule there are alleles inherited from mom, from your dad's mom, and alleles inherited from your dad's dad. And this is the only time that the maternal and paternal DNA molecules, chromosomes, actually combine and can be on the same DNA molecule. So crossing over can occur here. Another important process that contributes to genetic diversity in meiosis I is called independent assortment. So I think it's easy to see that I've drawn the pink chromosomes on one side and the blue on the other, but I could have just as easily drawn the big B, the little b chromosome on this side. And those two events are expected to happen with equal frequency in different meioses. So you can see that there are different ways to align these chromosomes in metaphase I. And this can lead to a huge, huge amount of genetic diversity. So you have 23 pairs of homologous chromosomes, and each one of them, each pair can line up one of two ways in meiosis I. So there are actually 8.4 million different ways that the homologous chromosomes can line up in you when you're undergoing meiosis. So 8.4 million different possibilities just from independent assortment. And that's not even counting the diversity you get with crossover events. So lots of diversity created this way. So in metaphase of meiosis I, the homologs align with each other on either side of the metaphase plate. In anaphase I, the homologs separate from each other. Telophase I, they're at opposite poles. And in cytokinesis, this cell actually separates into two daughter cells. So all I'm going to draw here is these DNA molecules on this side of the cell in one cell, and then the other ones in the other cell. And for simplicity, I'm not drawing the result of crossing over. So after meiosis I and cytokinesis, we have two daughter cells. And let's count the number of chromosomes in these cells. So we started out with 4 chromosomes, and each daughter cell, notice, only contains 2. So we've got 2 total chromosomes in each cell. Each chromosome still consists of 2 DNA molecules. So notice that we've gone from a cell with 4 chromosomes to one with 2. So we've taken a cell that's diploid with 2 copies of every chromosome and made 2 haploid cells. Because we've reduced the chromosome number, meiosis I is called a reduction division. So next, in meiosis II is going to look very, very similar to mitosis. What's going to happen is that the chromosomes are going to align single file in the middle of the cell in metaphase, and the sister chromatids are going to separate from each other in anaphase. And I'm going to draw the result of meiosis II in cytokinesis. So all I'm doing here, I'm going to separate these sister chromatids, and each one will end up in a different cell. So notice now that each cell still has 2 chromosomes. These cells are still haploid. We have not reduced chromosome number, but we've cut in half the number of DNA molecules. We started out with a cell with 2 chromosomes and 4 total DNA molecules, and now we have cells that have 2 chromosomes, each consisting of 1 DNA molecule. So this is the result of Meiosis II and cytokinesis. So what we have here are 4 haploid gametes, and nothing happens to the DNA in between the time that meiosis ends and the sperm forms. So all that's happening is that the cell is maturing into sperm. And notice that each of these sperm have 1 copy of every gene. So what happens at fertilization is really very simple, is that the sperm unites with an egg. So this was made in your mother via the same process of meiosis. And let's just say in her egg, the egg has the genotype big A, little b. So all that's happening at fertilization is that these cells join. And when I say cells, all I mean is that the cell membranes are fusing. The DNA itself is not going to fuse in any way. So what we end up with at fertilization, after fertilization, is a zygote. In this zygote, all I'm going to do is put these DNA molecules together here. This zygote has the genotype big A, little b, and this could be you. This zygote just divides by mitosis multiple times to give rise to a multicellular organism like you. And each cell of your body has these chromosomes and these particular alleles.